Hello everyone, my name's Grace and my project is understanding the interaction of symbiosis and growth in the temperate coral Astringia poculata, and I did this project with Dr. Randy Rochin as part of the Rochin Marine Ecology Lab. And the coral you see pictured here on the screen is Astrangia poculata, and we're studying this coral because it has just one fundamental difference from tropical corals that makes it really a useful organism to study fundamental coral processes like symbiosis, and I'll be unpacking that today. So Astringia is a temperate coral. It's not a tropical coral. It lives along the coast of North America from Cape Cod to kind of the Gulf of Mexico area. And if anyone has swam in these two bodies of water, they are very different temperatures. So Astringia is a very temperature resilient coral, which makes it useful to study lots of different species of corals because it has such a wide temperature and geographical range. And where tropical corals are really constrained to the warm water, this is not true for Astringia. Another factor is that Astringia has an algal symbiosis. This is, again, true for all corals, especially tropical corals, because they absolutely must have this symbiosis. The coral and the algae like to take care of each other, so the algae performs photosynthesis and gives all of the energy and the sugars to the coral, and in return, the coral gives it a nice little home place to live away from the dangers of the open ocean. But this process is kind of breaking down due to global change. High temperatures can cause this process to become not so beneficial. So in stressful conditions, when water temperatures are too high, the coral will want to expel this algae because it's just too stressful to have in that system. And this causes the bleaching that I'm sure everyone has heard about. But it's really hard to study the effects of bleaching in a coral that is stressed. When tropical corals don't have this algae, they're stressed, and so we can't study this system because the coral is not behaving as it usually would. And this makes Astringia really, really important because Astringia can exist with and without its algal symbiont, but it's not stressed in either condition. This is totally normal for this coral, and it makes it really useful to study this algal symbiosis. And so what we know about this coral so far is in terms of symbiosis is how it affects growth after the coral has been wounded or maybe a fish has taken a bite off of the tissue. We know how symbiosis affects survival in climate extremes like high temperatures and also intense plastic pollution. As you can see, microplastics in the coral right now. We also know how symbiosis can vary as a factor of seasonal variability, and also how that can influence growth. But these are all how symbiosis behaves under changing stressful conditions. We don't know how this organism just lives and maintains the symbiosis in very ambient conditions. So that's what we want to figure out, because if we can understand that symbiosis better, we can apply it to more pertinent situations like tropical coral bleaching. And so this is our main question, is how does this animal in symbiosis function without extreme stressors? And how we did this, how we answered this question, is we collected corals from Narragansett Bay in Rhode Island across a range of symbiotic states, so aposymbiotic, meaning it doesn't have any algae inside of it, and symbiotic, meaning it does. And we ran a 5-month experiment from September to February, and we manipulated 2 environmental conditions, light and food. And why we did this is because we wanted to see if we feed the coral versus feeding the algae, how will that affect the symbiosis? So light is the food for the algae, it's photosynthetic, it can make its energy that way. And then food, we used brine shrimp in our study, is the food for the actual coral animal. So how will combinations of food for the coral and the algae influence growth and symbiosis is what we wanted to determine. And we did this by taking monthly photos and digitally quantifying surface area, number of polyps, and a polyp is just one of those singular circle tissue areas, and also how they would influence symbiotic state. Surface area ended up not being significant at all, so I will not talk about that today. But some of our results were actually exactly what we expected. So symbiotic state changed significantly. They all started out about the same, having the same symbiotic density. And the light conditions, which are the green and the yellow lines, increased in symbiotic state significantly. And this makes sense. They're available with light, and light helps the algae grow. And so it just makes sense that with light, something with photosynthesis would grow, and that's what we saw. And similarly, in the dark conditions where there was no light, there was less and less algae over time. The symbiosis broke down. Again, this is not stressful for this coral, but it is useful to see that light is a very big factor in determining symbiosis. Food also did have an influence, but not as big. As you can see, the rates at which the symbionts were gained or lost did vary over time as a result of food, but not significantly. What we didn't expect was this. Our growth was the exact same almost for every single treatment. We thought that if you fed the coral and you fed the algae, it would just be in paradise with all of this nutrients and it would grow super fast compared to the other treatments. And if we removed those in the dark starved treatment, which is the red line, that this coral would be so stressed out, it would not have fun, and it would just not grow at all or even lose polyp numbers. But we found that every single treatment lost polyp number, which is not what we expected. So either symbiosis is not as important as we originally thought for growth, or it's just way more complex than we thought, and we haven't teased out those complexities yet. And so some future directions to help tease out those complexities would be to quantify the actual growth rate and the rate at which the symbionts leave or come into the coral. We can also measure the actual symbiont density in the coral by counting the algae cells. We just didn't do that here for time. And then also we want to know specifically which factors are influencing these rates of change the most. But overall, we know that this symbiosis is really, really important. But the fact that these corals are responding so similarly, no matter the symbiotic state or the feeding conditions that we put them into, really challenges the assumption that this is a mutualistic symbiosis in which both the coral and the algae benefit. There have been some studies that suggest in some conditions the algae might actually be like an energetic parasite that's harming the coral, and our results kind of support that. And so despite a little bit of uncertainty, we were still able to unpack the fact that this symbiosis underlies all corals, and it'll be increasingly important to understand how tropical corals will respond to climate change by solidifying this symbiotic relationship. Thank you.
Any questions?
This is so quick and probably obvious, but can you explain what symbiotic density is and how it's measured?
Yeah, so symbiotic density is the actual count of algae cells that are within the coral tissue. And so we can measure that in a bunch of different ways. The reason why we didn't physically measure it here is because it's destructive. You actually have to kill the coral and break open its tissue. And then we can put the tissue on a slide and actually count the individual number of cells, per water volume. But how we did it here is by taking our photos, we can put them into a program on MATLAB and outline the sections of the coral that are interesting to us. So that would be the tissue. And that program will give us basically a color value. It tells us how red the coral is, and we can use that as a proxy for how dense the symbiont is in the tissue. Yeah.
Thank you, Grace. That was fantastic. Really, really interesting. Can you comment on, when I think of a person losing food, I don't think I'm going to last for a particularly long time. What about coral? How long does it take coral before, if you starve it, is there something about its life cycle and how long it lives? How does that factor into your study?
Specifically for tropical corals, I'm not sure, but at least for Astringia, they're known to, at least in the winter periods when food and plankton and that sort of abundance dies down, they don't have the opportunity to get as much food. So this species is known to go into a period called quiescence, which is just like a dormancy. It's like hibernating almost. I'm not sure if this happens with a bunch of other species, but at least for Astrangia, it's known to just really, really shut down its metabolic activity so that it can make it through that winter period that's stressful and cold. But in the summer, when there's more food and more light, then it is able to get back into the swing of things, per se. Yeah.
Okay, I have one quick question also. So you kind of touched on this a little bit at the end, and this is a little bit of a disorganized question, but like, can you explain a little bit more insight into like the timeline of this coral and the symbiotic relationship that the coral and the algae had, and if potentially this relationship could be becoming something that's not symbiotic anymore due to global warming and microplastics, like both
Yeah.
are trying to survive?
Yeah, so specifically in tropical corals, when the water pH becomes too acidic or when the temperatures become too high, the coral can't take care of itself and take care of the algae. So it says, for the time being, I'm just going to expel you and we're just going to live like that for a little bit, but it can't live that long for like months on end. So eventually, to survive, it will have to acquire that algae again, and that's when bleaching becomes really detrimental, when the conditions like high temperature stay high for a really long time, and that's why it's concerning to us. But as far as the timeline in Astrangia, they can kind of exist without the symbionts for a really long time. Some of the corals that we collected were without algae from the beginning and stayed that way throughout all 5 months. But I'll go back here. The reason we think that this might be breaking down is, if you can see in the yellow line, that treatment got light but no food. So we're thinking that if the symbiont or the algae is being fed, but the coral's not, it might be too energetically like taxing to hold on to that coral. The coral might be expending more energy housing the algae inside of it than it is taking care of itself. And so that's why we thought maybe it's hovering around a sweet spot here, as it just kind of stays around the same symbiotic density. But I hope that kind of answers your question.
Thank you so much.